All right, so what we've got so far is we've got the RAG protein that's regulated. It's regulated. And so when there's leucine present, the RAG protein, the GATOR2 is free to inhibit GATOR1, and the RAG is GTP loaded because the regulator, it acts as a GEF to it. So now we've got a GTP loaded RAG, and that's great, but we still have to bind it to mTOR. And so what we think is happening is that's great. You've got the RAG protein is GTP loaded, but it's not like it's going to act as a magnet and all of a sudden everything's going to come to it. So wouldn't it be cool if you could take that lysosome that's now got an active RAG and move it around the cell like you have a Roomba? Well, the Roomba is actually regulated by VPS34. And this is the vesicle protein sorting molecule, the 34th of these. And it's a kinase, but it's one of the oldest kinases in our cells. And what it is it's a class 3 phosphoinositide 3 kinase. So it's like PI3 kinase, but it's slightly different in that it's going to phosphorylate the 3 position of the inositol ring to make PI3P. PI3 kinase makes PIP3. This makes PI3P. It's a slightly different structure. Just know, all I want you to know is that VPS34 is an inositol kinase. And so it's going to phosphorylate the 3 position of the inositol. So it's going to make, actually, what it makes is it makes PIP instead of making PIP3. So this is just PI, and that's the 3rd position phosphate. So it's phosphoinositide phosphate. So this is PIP1. Okay, whereas PIP2 becomes phosphorylated by PI3 kinase to make PIP3. This is PIP1. Okay? It's the human homologue of a yeast gene that's involved in moving vesicles around the cells. And it's permissive to the activation of mTOR, likely by moving Rab-positive membrane or lysosomal membranes where Reb is located. So the idea here is that when leucine comes in or some other amino acid comes in and activates VPS34, VPS34 can phosphorylate lipids on the lysosome, and what that does is it actually increases the movement of the lysosome. That's the idea. And so when it does that, now if you've got RAG protein that's been GTP loaded and you've got active VPS34, basically you've turned on the Roomba, and now you can suck up all of the mTOR throughout the cell. Interestingly, if we look at after exercise, 3 or 6 hours after exercise, what you can see is that VPS34 activity goes up, and it peaks around 3 hours, and it starts coming back down later. So it's one of the things that becomes active after exercise. And we later showed that this activity correlates again with the increase in muscle mass and strength, and VPS34 is activated by leucine. So the idea here is that leucine comes into the cell. It's going to bind to cestrin, and it's cestrin 1, 2, or 3, it doesn't matter. Cestrin 2 is no longer going to be able to bind to GATOR2, and GATOR2 is going to be able to inhibit GATOR1. And when it does that, here's the RAG protein here, and it's bound to the regulator. So when GATOR1 is inhibited, there's no longer that gap protein. The regulator can serve as a GEF and GTP load the RAG-AB. When this is GTP loaded, now it can bind to Raptor. Okay? So here's the other thing that's happening. Leucine is coming in, and VPS34 is going to these Rab7 positive membranes, and it's phosphorylating phosphoinositide. And it's making this phosphoinositide phosphate, or PIP1, and that's actually going to increase the movement of the lysosome. So now we have the ability to bind to mTOR, and we're moving around this structure within the cell. And now what it's going to do is it's going to quickly vacuum up all the mTOR within the cell. And look what happens. Now you've got Raptor right here. We showed you that on the Rab7 positive membranes is where you find Reb. And if we've had some sort of stimulus to move TSC2 away, Now Reb is active and mTOR can be fully activated. And PRAS40 shouldn't be there, but you understand what's going on here. Now we can phosphorylate things because it's fully active. This is when you get complete activity. All right, so now what happens when we combine resistance exercise and amino acids? Here's our unknown sensor going into RIC, our really important kinase, which is phosphorylating TSC2 and moving TSC2 away from Reb. Reb is now going to become, there's going to be a GEF that nobody's identified yet that's going to move GDP out and allow Reb to grab GTP. At the same time, leucine comes into the cell, activates VPS34 and starts moving things around, but it also binds to cestrin, which causes it to release GATOR2. When it releases GATOR2, GATOR2 can now inhibit GATOR1, and GATOR1 is normally the gap protein for RAG-AB. When there's no gap protein, the regulator can cause GTP to be lost, or GDP to be lost, GTP to be gained, and now you've got an active RAG protein, an active Reb protein, you bind mTOR, and now PRAS40 would quickly get moved away because the really important kinase would phosphorylate it, and now you've got highly active mTOR. Because really you need both signals. You need to get rid of TSC2 and turn on Reb, or GTP load Reb, and you need to have the leucine to get rid of cestrin and allow us to block GATOR1 in order to GTP load the RAG proteins. So basically what I'm telling you is all of our ability to grow our muscles after resistance exercise and eating protein is down to a small number of small GTPases that are localized in such a way as to activate mTOR.